Assalamualaikum, I am Nur Irdina binti Nozri with the metric number of 146806 from Bioprocess Division and from School of Industrial Technology. I am going to present you my lab practical video 3, which will cover the topic of aseptic technique, preparation of culture media, handmade, and thawing cell. Enjoy watching! The purpose of aseptic technique is to ensure no contaminations can occur to the animal culture cell and to minimize the chances of being contaminated by highly contaminated cells that we are working with. The chemical and equipment involved in this technique is 70% ethanol, Kim wipes, and lamina hood. The first procedure involved in this aseptic technique is wear an appropriate lab attire such as lab coat, gloves, goggles if needed, and mask for our safety purposes. Secondly, turn on the UV light from lamina hood for at least 20 minutes before proceed with our experiment. Thirdly, wipe our working area with 70% ethanol with Kim wipes and ensure to wipe all areas. Wipe. Fourth, we have to autoclave all possible materials and chemicals before we use them in our experiment. Fifth, arrange the chemicals, beakers, equipment involved for the next experiment in an ideal position or in a way that we are able to do our experiment smoothly. Lastly, waste materials must dispose in a suitable disposing place. The second video I am presenting is the preparation of culture media handmade. The purpose of this technique is to prepare a handmade culture media in powder form, which has its own benefits than using a prepared media. The chemicals and equipment involved in this technique is sodium bicarbonate, ultra pure water, fetal bovine serum, and antibiotic, serological pipette or measuring cylinder, a large beaker, and a large blue-capped bottle respectively. The procedure of this technique involved: firstly, we must perform the aseptic technique as before we proceed with our experiment. Secondly, the culture media is dissolved in ultra pure water and 2 grams per liter of sodium bicarbonate is added in a large beaker. Then, gently mix the mixture. The ultra pure water is then added until it has reached 445 ml. Then, the pH value is adjusted to 7 to 7.3 by using a pH meter, which is a suitable range of pH condition for animal cells to grow in. We wanted to make a 500 ml of media, therefore 10% of FPS with an amount of 50 ml and 5 ml of 1% antibiotic is added into the large beaker containing 445 ml of media. The 500 ml media is then filtered by using a pump filter to fasten the process of filtering. This step is crucial to remove any insoluble particles or lumps in our media. Next, the media is then stored in a 4 degrees Celsius refrigerator. The last video I am going to present is thawing cell. The purpose of this technique is to enable a frozen cell to viability or thaw the cell again in order to culture the cells according to our experiments. Plus, thawing cells is crucial to ensure the culture can be recovered quickly. Bear in mind that this technique must be done quickly yet carefully because the cryopreservation composition in the cells can be toxic to cells after the thawing process. The chemicals and equipment involved in this technique is falcon tubes, micropipette tips, flask, incubator, and water bath. The first step of this technique is to perform the aseptic technique and ensure that the media is warm enough by putting it in a water bath. This is to ensure the growth of the thawed cells with room temperature. An important remark is to wear proper attire when handling liquid nitrogen as it can cause skin irritations or can be potentially hazardous. Secondly, the thawed cells are removed from the liquid nitrogen into a dry ice, optional because the cost of dry ice is expensive, and we put the vials in a water bath. Thirdly, 5 ml of thawed cells were then transferred into falcon tube by using micropipette and 10 ml of media was added into the falcon tube. Then, the supernatant in the falcon tube is removed and the remaining cell pellet was then resuspended with media by using micropipette with a motion of up and down in the falcon tube. Next, the culture was moved to a flask and incubated in an incubator. An incubator's temperature is adjustable to our desired temperature according to one's experiment.